This is Christina Bauer, the Texas Lyme Alliance, and I am here with Dr. Jay Rajadas.
Hello.
Anything that you hear in this video is not medical advice. It is the discussion of us personally, Dr. Rajadas and myself, and always use your physician as your guide. We're definitely not recommending disulfiram. That is between you and your physician. Hear from the expert on his take of disulfiram for Lyme. So Dr. Rajadas is the founder of BioAd, which is a lab at Stanford University. And
Drug delivery person. My PhD is in structural biology.
okay,
So I understand the therapeutic molecules, how they organize itself, how it is delivered,
okay.
how it is hitting the target. These are all my area of expertise.
Oh,
I'm
we wanna
also an
mention that.
adjunct professor in UCSF.
Yeah,
Yes.
I think
Okay,
that,
we came to see his presentation at the Live Lyme Foundation, and it was phenomenal. For 2 days, I loved all of the information. The presenters were wonderful, but you really stole the show. And we wanted to come today and interview Dr. Rajadas because I think his groundbreaking discovery of disulfiram in treating Lyme is so amazing.
Not just me.
Right.
So it's a team like myself and the scientist, originally Dr. Ravindra Pothineni, and you know him, I
Yes,
think.
thank you for
And
mentioning.
also currently Dr. Harihara Potula, he is also working on it with a passion. And he's also focused on understanding the immunological aspect of disulfiram therapy. And we have another person who is basically a formulation scientist who is interested in developing a proper formulation for disulfiram. His name is Dr. Muhammad Inayatullah. And then we have another neurologist who is interested in studying the neurological aspect. His name is Rayafuddin Ahmed. And I also mentioned about Dr. Kwon Min Kim. Kwon Min Kim is interested in studying how exactly the borrelial infection is affecting the signaling, actually, how it is affecting the neuronal function, the cognition, both in an animal model and also in a chip. In fact, as I mentioned during my talk, this paper appeared in the early this year in Journal of Neuroscience with a cover page.
That's great.
So this is the great team I have, and I think they are all very passionate about what they are doing.
Well, I wanted to get their name on film so that the Lyme community could just hear how your team has come together and all of the different things that they're working on to help bring a cure for Lyme disease. So please thank them for
Thank
us.
you.
And
In my lab, we have a program not only to screen the drugs for killing the persisters of Borrelia, we also have a program simultaneously what the Borrelial infection is causing to the immune system. So we have an immunologist in our lab who is just taking care of it.
that's wonderful.
Yeah, we also have a neurocognition team. There are two people. One is senior scientist. His name is Rayafuddin Ahmed. And another one is Dr. Kwon Min Kim. Both of them are focusing only on the neurocognitive issues of the Borrelia infection
That's
in
wonderful.
our preclinical model. And currently, they are going to be engaged in trying to find out the disulfiram intervention, what happens to the cognition. Whether the intervention during the treatment time, the cognition is improving or it is kind of going the other direction till the treatment is over. Because this kind of message, this kind of knowledge, this kind of understanding is extremely important for the future physician who is likely to use this therapy after the clinical trial, after we get an FDA approval, after we get the proper, the data, we just analyze the data. Of course, we also understand clinician are using this currently, but it is better we just to do few clinical trials and then try to have a data with the placebo controlled randomized clinical trial. So it's not that he said, she told, not like that. Rather, a solid data.
Right, that's what we need.
Yeah, that's
Yeah.
what, yeah. I really think at one point we will raise money, enough money for us to complete the clinical trial. That trial, the results of that trial will answer all most of the questions, I think. But again, I always believe this when the trial, clinical trial is going on. Yeah, basic scientists like us need to support the trial or even ongoing trial with a lot of simultaneous research and also very focused, not just a hypothesis driven results, but just hypothesis driven experiments, but just an observation. Try to teach us what's going on in the mammalian system when these things they together they act, whether there exists a synergy between our immune system and also the disulfiram therapy, whether there exists any synergy between neurological aspect of it, and also so we just wanted to figure it out how exactly this particular therapy is going on to because when we developed this, this is as you know, it's 2016 paper. We started from the beginning with the high throughput screening. We tried to find out the best molecule that can clear the persister. That is why we got into this. And this is the selection actually. The even though several molecules we got to the better hit, Ravi did the selection actually. He ranked the thing. If you see in that paper, we ranked
I saw
disulfiram
that.
first, and and then we started trying to do more experiment, and then wanted to get the best out of the thing because we always believed one molecule, not two or three, because it's complicated the therapy, and also it also the drug compliance is a problem, approval is a problem. So we wanted to avoid all those things. We thought we will try to find just one molecule. As you know, as many people think, Lyme disease is a very complex disease. Even people are questioning the cause for this chronic condition. Nobody is having a proper agreement about what's going on. But I always tell people that whatever it is, uh, pain is real, and it is let us do something, let us try something. So that the famous hypothesis which originally Monica Embers, she in her lab work, like she showed, in spite of an antibiotic treatment in her primate model, she showed the existence of this bacteria we call right now, we are calling them as persisters. So we asked this question, how we can develop something that will sterilize the persisters, that will clear the persisters. And again, in that disulfiram kind of ranked number one. We have molecule other than disulfiram, as I mentioned during my talk, azulusulin and another one. It's a kind of antibiotic, but disulfiram is kind of slightly different from azulusulin, even though we always recommend, for a development purpose at least, azulusulin for an acute Lyme, and for a persistent and chronic condition, we recommend disulfiram. We wanted to develop in that way, not because it's our choice. It is because when people show up with the early, infection within few months of their infection, if they wanted to show, go and then get a medical advice, it is better, they get the prescription of a kind of typical antibiotic.
Right.
And we showed several times with our publication, and also we repeated several times azlosulin is number one in that. So regarding disulfiram, back to disulfiram, disulfiram is an amazing molecule, and in fact, I was telling my lab mates at one point it will be selected molecule of the year because it's so much wonderful things are coming out of it.
Yeah.
Because this is a very very interesting molecule where it takes care of sterilizing or killing the bacteria. At the same time, it is also addressing the inflammation that is triggered by this bacteria. In our several years of experience, our experiments in preclinical model, we always found out this bacteria, it like in the way cancer cells survive, this bacteria they propagate through the inflammation trigger when they move around. So they trigger the inflammation and then they move around and they survive with that. That's what we found out. Is the top 20 molecules we published in 2016. That is our select candidates. But these two kind of, with a lot of experiments, and not only that, now we are focusing on some molecule which is outside this 20 top hit also. But these two molecules, with our experience of this is kind of the best what we have.
Wonderful. Well, we're so grateful that y'all did that work, and that was funded by Bay Area Lyme.
Yeah, originally by Bay Area Lyme Foundation. In fact, they started funding as.
This is a really important discovery for the treatment of Lyme disease. How do you think this treatment is going to affect the Lyme community?
I think it's too early to come to any judgment. So initial, the preclinical data, the signaling, whatever we see, it looks very, very promising. And whatever the toxicity which patients are reporting in their social media, we don't see that much toxicity in our preclinical model. It could be due to species variation, the way rodents respond to the infection, and also the therapy is slightly different from human response. But thank you very much for putting all this experience in a social media. This is kind of amazing experience for us. We learn a lot from that, so that we can ask the right questions in our experiment preclinical model. So what if we just do this particular design, experimental design, what kind of answers we are going to get, and how this answer is going to help ultimately the user end. This kind of experience is very very important. Basically, I always think either it is social media, the data, or a conversation, informal conversation with patients sometimes, and sometimes with the clinician. I always think it's a data. Anything, a keen observation is a data. That's why, when I started the presentation, I started with the aspirin drug development.
Aspirin.
Even aspirin, if you know, it is almost 3,500 years people are aware of the therapeutic effect of the willow bark. People used to make soup, they used to make extracts, or and then they take it, and missionaries, Christian missionaries, used to help people using that. But nobody did any scientific investigation trying to develop that as an organized therapy. So that is what I think we are doing in a laboratory. We try to find out this kind of anecdotal evidence, like what you and other people are reporting, trying to make a very good scientific investigation, an organized scientific investigation, and try to ask the same question you are reporting, making the observation, media observation, and transforming them is a scientific inquiries, and then ask those questions, and then try to get an answer from our preclinical model. Then later, what we get out of it? We get out of it a mechanistic understanding how this particular therapy works. In case if you're successful, in case if you're successful in getting necessary funds, and then be successful in all our endeavors of trying to understand how exactly this therapy works, whether persisters, sterilization is extremely important to for the Lyme patients to become totally all right, or we need to do with one molecule, try to eradicate these infections, at the same time try to treat them with the same molecule, the inflammation that is going on, ongoing inflammation. All these questions, we will get an answer in our pre-clinical studies what we are currently doing. Much more important, we wanted to understand, using this basic technology, try to understand how exactly why uh, uh, chronic and also persistent Lyme infection in mammalian system ultimately end up so much cognition issue, so much neurological issue, so much immunological problems, why it is happening. This kind of questions we wanted to find an answer, and it will be lovely to find out whether molecule like disulfiram can intervene all of those issues. It's kind of too much to ask from one medicine, but I think if it happens, it happens.
And it's broad spectrum from what we see, right?
That's correct. That's correct.
And so, to answer the question, that was a really great way how you are listening to the patients, and patient-led information is giving you data for what you see in the lab, and then you are using that information to help us back in return. What do you see as far as copper toxicity?
That's a great question about this copper accumulation, we call it. In fact, we also worked previously, I published a couple of papers using another copper chelator, very good copper chelator like disulfiram.
Okay.
Be used as a therapeutic agent. So when we were using, I asked my graduate student, this is my previous lab, not Stanford lab. I asked my graduate student, okay, we are giving this therapeutic, molecule. It's very good copper chelator, and why not you just check the brain accumulation of copper? I asked her. She came back, and then with a big smile, because she thought I predicted there will be a copper accumulation in the brain. She said, yes, there is the copper. And unfortunately, we didn't publish that work. So, yeah, when you have a, like molecule like disulfiram, which got a very good blood-brain barrier, blood-brain barrier penetration, and also it is covalent molecule. That means the disulfiram gets in, it's likely to get fixed in the sulfhydryl groups, cysteine groups, that is, and so you will be having lot of disulfiram molecule covalently linked in the brain. So that means the brain capability of retaining copper will be very high. So I don't know how much problem that will cause. Obviously, the patients, earlier the anti-abuse, people who are like years they are in disulfiram, nobody came back and then told developed a lot of copper accumulation in the brain or all other organ. So far I know not that much data. Maybe there exists some data which I would love to learn about it if there existed data. But Lyme patients, or Lyme animal model, what we have, they are slightly different. They have as I told you that in their brain we find, and other people also showed, they have perpetual inflammation going on even after acute exposure of Borrelia in these animal models. If that is true, then their brain absorption of metals may be different from normal people. People who are using it for anti-abuse, maybe they are different. Maybe that is the reason they are maybe they are like, I don't know. Unless we ask those kind of questions, we can never answer. The best thing is, that is my priority question I'm going to answer. Like, we wanted to take animal models and then expose them with Borrelia, develop a possible, accelerated chronic model. Recently Dr. Zhang developed, and using that model and then treat them with disulfiram, and then try to see whether all that Borrelia load is going, whether they totally getting sterilization. After they get the sterilization, the first question we wanted to ask, we wanted to measure how much copper is accumulated, and trying to find out whether disulfiram is causing any brain lesions because of this copper accumulation. Whether we can interpret whatever the side effects people are reporting because of disulfiram, whether we can translate it into a very good, discernible scientific uh, project. That's all we are planning to do, and we are getting more and more excited about, wanted to love to understand how exactly this therapy works. Because this therapy, which we think, if we understand properly, there is a possibility in future it can morph into much better and efficacious
That's
therapy.
our hope too
Yes,
as a patient.
yes.
What do you think as far as azoxystrobin, you had mentioned as a penicillin antibiotic? You were saying that it's best for acute Lyme, but when a German friend did some research for us, he came back and said it's $4,000 a month. It's only NIVs.
Okay.
Okay, as far as we found, do you still think it's far superior for acute than disulfiram? Would it be worth us trying to get that therapy, and how long would that therapy last?
So the cost, actually, it is the why it is so expensive in Europe. Of course, it's not even available in US. Why azelastine is very expensive? Because there is no demand for it. There are so few people are using it. Let us say that there's going to be like thousands and thousands of people are going to be using it. There will be a company, procuring it and formulating it and manufacturing it. The cost is going to come down. And again, always IV therapies are very expensive. And here, so this 4,000 is not going to exist as 4,000. It's going to be much, much less than that when we are, when we have a demand, not just
For
a
it.
rare demand. So people are, I don't know why exact, for what purpose some clinicians are still using azelastine. It's an IV, and to have an IV therapy, and we are talking about oral therapy. So in our lab we found out the way to make it, yeah. So uh, it's likely to be much cheaper.
That's
Than
wonderful to hear. I can't imagine. But the duration, are you thinking, of course, this is hypothetical and you're not making advice for recommending this to other people, but do you think it's going to be a much shorter course than what patients are experiencing with being on antibiotics, for instance, combo antibiotics, titrating antibiotics for years on end? Do you anticipate that something like that would wipe this out very quickly?
Yeah, this one, it's not, I don't think it will work great for chronic
Chronic.
condition. Chronic condition is more complicated condition. As you all know, so much debates going on. What's the cause for this chronic condition? But acute is kind of straightforward. People, they see, not straightforward. There also diagnosis is a problem, but at least proven case, let us say 60% of the patient, they have very good proof that they have acute exposure of Borrelia. Those patients, I think, azulocilin is the best. It will be a best option for them. Because, as I told you earlier, it is an antibiotic. Kind of, we have a lot of data to show it is not having any toxicity, and interestingly, no side effects so far.
Oh, that's wonderful. I
Yeah,
didn't know that.
it's not unlike the disulfiram. There is no expected side effects
effect.
because people used it in a very heavy. Even babies they were exposed to azulocilin.
Oh, wow.
Yeah, in fact, different routes of delivery people tried. Even I saw a paper where it talks about people delivered to the lung, babies. Because those days they used to use it to clear the infection in cystic fibrosis. Yeah.
Is
Yes.
it past the
So
brain barrier?
I was told we are yet to do that experiment. I was told it got a very good blood-brain penetration
Penetration.
also.
That's wonderful. Okay. So, as far as disulfiram being the best choice for chronic patients, is that because chronic patients have way more inflammation than acute patients, and so therefore they need anti-inflammatory therapy?
Yes. So again, I'm not a clinician. I can't
Yes, and
tell
of course
anything.
we're not, but we're
Yeah.
speaking
So
hypothetically.
hypothetically, and based on our preclinical work, we really think for chronic patients, patients with chronic condition, disulfiram will be better because it addresses many things at the same time.
So it's broad spectrum, but it's also helping to dampen that
That's
chronic
correct.
inflammation.
Yeah, that's correct. That's correct.
But what do you see in the future for Lyme patients and disulfiram? You have done the mouse study, and that proved in your slides that it also eradicated disulfiram
Yeah, so
eradicated in mouse study.
we have we are yet to we just submitted that paper for publication. It is still in the review. Yeah,
Okay.
we do have data to show it is sterilizing in animal models. All complete the preclinical work and then somebody do, yeah, placebo controlled randomized a clinical trial, and then after finishing the clinical trial you get a data, and based on the data the clinician need to decide the course of action and then developing this as a therapeutic options for a chronic condition, and that includes azulocilin also. So there need to be a phase 1 clinical trial and phase 2 clinical trial, as it is always do no harm. That is the, that always clinicians take that is very seriously, and this kind of systematic and scientific way of developing any molecule addresses that do no harm issue very well, I think.
Seen it in vitro
And also in our animal model.
and in vivo.
Yes.
And the percentages of clearing Borrelia have been what?
So our experience, we really think we understand the slow delivery issue. We understand the optimum delivery modalities and also the amount of drug concentration that needs to be present in this rodent model. We think we have seen good, very good clearance, sterilization of this Borrelia in our models. But again, there are so many questions need to be answered. Whether such a sterilization happens in human, that needs to be answered.
Right. Yes.
Yeah, that kind of answer will happen only in a controlled clinical trial, not like
Mouse
just
study.
yeah.
So have you, looking forward, have you scheduled that clinical trial
Yes,
in
we
human
are.
studies?
We
That's
just,
the important
yeah,
question.
we just got a small gift to do the clinical trial, and ultimately there is a possibility that we can develop into a bigger
Clinical trial.
clinical trial on that. Appropriate amount of money to given to a researcher like me, we will ask right questions And right questions always come back with a proper answer. Those answer will be very useful for therapeutic development. And as I told everybody in the meeting today, the ultimate goal is better delivery of disulfiram, because I'm a drug delivery person.
Right.
And better delivery, better formulation of disulfiram with least side effect possible for this set of patients. And if possible, with these resources, develop something much better than
Easier
disulfiram.
for the patient.
Yeah, much easier or better disulfiram analogs, or a different molecule which addresses the same mechanism that we can. That is what I will do if an appropriate funding is given to my lab, because excessive funding, I personally felt, just because somebody is having like $40 million or $50 million, somebody come and then give such a big money to a researcher. I have never seen that morphed into a good therapy.
Right.
It is always with very focused, planned research with an appropriate support.
Exactly. That's what's so important, because from my research, there's been $32 million so far given to researchers for Lyme disease research, and we're still wondering what is the best therapy. We don't have protocols that help us. I see that a person like your lab has dedicated, I think you said 39% of your entire lab toward...
Yeah, it's around 40% of the people are involved in developing therapeutic molecules for both acute and chronic Lyme disease.
That's pretty impressive. And I just wanted to make sure the public audience hears that you had told us that your goal is to not stop till you find a cure for Lyme disease.
That's correct.
That's very
amazing. Our team is very passionate about this research.
Yes.
We love to be we wanted to participate along with anticipating, clinicians and other people, and of course, we wanted to participate with people like you, who really promote this research by, doing this kind of presentations. And we wanted to be participative people in the whole thing. Actually, I really think with this all kind of input and also proper support, there is a possibility we will come with the proper cure at one point.
Well, that's so promising to hear. It's just affected so many people on so many different levels. Thank you so much for your time for all of your effort. And from the Lyme community, we thank you so much for your efforts to bring a cure for chronic Lyme disease.
Thank you very much.